Hello everyone, my name is Darla Saunders, and on behalf of the Atlantic Salmon Conservation Foundation and the Canadian Rivers Institute, I'd like to welcome you all to today's webinar. This series is made possible in part thanks to the contributions of the Government of Canada. We are very delighted today to be hosting Wendy Monk. This is possibly our most highly anticipated webinar that we've had yet, and so we're really pleased to have you all with us today. Wendy is a visiting research fellow with Environment Canada, working at the Canadian Rivers Institute and the University of New Brunswick. We've got quite a few folks with us today who this is their very first webinar, so we'll go through a couple of housekeeping matters before we begin. Questions should be saved until the end of the presentation. To ask a question, you can use your webinar control panel, which is the little gray box that you should be seeing in the upper right-hand corner of your computer screen. If the box is minimized, you can hit the orange arrow and that will enlarge it. If you're using the audio of your computer, you can raise your hand, which is the little yellow icon that you should be seeing with a green arrow, and Michelle will unmute you, and then you can just ask a question directly. Or you also have the option of typing in your question on the control panel, and we'll read it aloud for you during the session. So I'm now going to turn the webinar session over to Wendy.
Thanks very much, and thanks to everyone for joining me today. It's kind of nice to know that so many people are so interested in what I'm going to be talking about. So today I'm going to start to introduce GIS tools within the context of kind of our watersheds and our river ecosystems. So just to start off, I thought you'd like to know a little bit about me. So I'm from the UK. You can tell by my accent, and I grew up just outside London, and I did both my degrees at Loughborough University, which is in the Midlands of the UK. And then in 2006, I moved to Canada, to New Brunswick, to take up a research position with Dr. Alan Curry at the Canadian Rivers Institute. And since then, I've been working with other researchers within the CRI. The background to my research is predominantly drawing on kind of large-scale patterns, spatial and temporal, and how they map onto kind of the aquatic communities within river ecosystems. And so I look at different rivers, I take different kind of large-scale climatic variables, land use variables, and I look at how those map onto the patterns and flows that we see in the river. So whether those are rivers within Atlantic Canada, but I've also done research across Canada. I then look to see how those patterns and processes drive the communities that we see within the streams. And primarily I focus on benthic macroinvertebrates. And when I take that information, I can develop different applied diagnostic indices to help us understand what's going on with our rivers and how we can use these benthic macroinvertebrate communities as indicators of potential stresses, for example, flow stress. Underlying everything that I do, though, is GIS, or Geographical Information System work. So my background is a geographer, and I'm kind of like a geographer meets hydrologist meets ecologist. And so I bring that knowledge that I have about geography into kind of the biological and ecological world. So where are we going today? In the next half an hour or so. I've got three main things that I'm going to try and cover. First off, I'm going to run a brief history of mapping, looking at both the importance of space and time, and how do we actually use maps today. Second, I'm going to look at how we disseminate information through maps. How do we share maps, and how do we present information so other people can understand it? And again, focusing on both the spatial and the temporal patterns. And then finally, kind of the meat of the presentation is looking at some of the GIS tools that are available to us. So I'm going to look at different programs, data sources, and also some applied examples from the work that I've been involved in. So this is my very, very brief history of mapping. In the figure on the left-hand side, which has the kind of like stone carving, that's actually one of the first examples of a map that's been found and unearthed. This is actually a map they dated it from somewhere between 500 to 700 BC, and it's from Babylonia. And it's basically the first thing, the first example of where people have tried to put down on paper, or in this case in stone, where they are and where things are in our relation to each other. Moving on several centuries, you know, in the early kind of late 1800s, early 1900s, we then start to have more precision and accuracy in our mapping. Of course, in the meantime, various kind of advances have been made in how we develop maps and how we understand both space and time. You can see the map in the middle there is actually a fairly good representation of a map that we see today. Of course, move on to now where we are now, and we have so much data at our fingertips. We have Google Earth, we can zoom in anywhere on the globe. We can take satellite information, we can have small-scale, large-scale information. And where GIS comes in is how we can start to get a handle on all this information and interpret it in a known and knowledgeable way. When we're looking to develop maps, there's a series of ways we can present that information. In the map on the left there, you'll all recognize as Canada, and so we can think about a political map, so where we are in relation to, let's say, the different provinces and territories. That's a known kind of geographical political map. The map on the right there is another way of kind of showing both space and times in terms of communities, but in this case, the person has actually taken information about different kind of online feeds and has put them into kind of a map. And you'd recognize that as a map, even though you know it's not a global map, and that information, the size of the countries, in kind of quotation marks there, represents kind of the amount of people using those online communities. And so it's another way that we can show information in a political sense. We can also have physical maps. So this map on the left here is a beautiful example from the early 1900s. And this is actually a section of the Mississippi River. And those different colored kind of swirls and things you can see are basically the meander channels, the historical meander channels from the Mississippi River. On the right there is for Fredericton and is from the Riverwatch website. You can see that this is a map of where there's been historical flooding. The 1973 flooding extent is in the red, and the 2008, the one that's kind of in more recent memory, is there in the orange. And this is where the kind of physical meets the human. So this is where physical patterns, in this case flooding, meets the human concerns, because we're concerned about where flooding is, and that may affect, for example, development. We can also think about things in terms of movement. So again, on the map on the left here, I had to bring in a UK example, but this is a map of the London Underground system, the tube or subway system. And you see, we use that, we can take that information, and as someone who's trying to navigate their way around London, they can pinpoint themselves on the map and navigate to where they need to go in a very simple and easy fashion. But we can also think about movement in terms of the movement of individual species or taxa. So this is an example here from Dr. Tom Linsari's work, where he was looking at the movement of different fish in relation to ice. So how do we use maps in freshwater science? I pulled together a few brief examples here. We can map our samples. You may go out and sample a reach, and you take your handheld GPS unit, and you can note down your latitude and your longitude. And then you want to plot where you took your sample. You may want to then start to extract information about your site and also your reach. So you could start to perhaps overlay some geospatial layers, and you could start to understand how your sample is in relation to the surrounding environment. You may want to use geospatial data in bioassessment, and it's one of the examples that I'll talk about at the end of the presentation, is how we start to use these kind of large-scale detailed information layers that we can now access to start to determine, how impacted our sites and regions are. Perhaps you want to start explore and quantify the upstream watershed. So you take a sample at a given point, but what's driving that? What's upstream of that site? And finally, as in the previous example, we may want to map target taxa. We may be interested in a particular species. We may be able to tag it and monitor its movement. Or we may be interested in kind of larger groups and being able to kind of target them and map their movement within the system. So we can think about ourselves putting our samples in a spatial context. So we've gone out to a river and we've taken a sample, represented here by the yellow circle. You can see that when we're taking the sample, often what happens is we take additional information while we're collecting our sample, but really that's just telling us about the adjacent land use, the adjacent riparian area. Maybe you can walk, 200, 300 meters upstream, downstream, across the banks. But what you're really interested in is what's going on upstream, what's driving the patterns that you're seeing at your sample point. So GIS allows us to do that. It means that we can then look at samples in relation to the upstream characteristics. We can also look at samples in relation to the other samples within the watershed by extracting that geospatial information. We can look at the individual watershed, or maybe we could compare to a similar watershed. So what GIS would allow us to do is you could take a sample in a different watershed, quantify various characteristics of the upstream catchment, and you'd know how similar your two watersheds are. Or if you were interested, you may want to compare to a completely different watershed, and so you may want to be looking at contrast in your patterns. We can also think about putting our samples in a temporal context. Maybe you're lucky and you've been able to go out and sample in multiple years, but that was so you get a different community in each different year. But if you're lucky, then there's geospatial information, satellite data predominantly, which are often taken in multiple years, sometimes multiple seasons throughout the year, and that allows you, again, to put your sample in not only the spatial but also the temporal context. So how is your watershed changing over time? And then how is the sample that you're taking reflecting that change? So this here is the hierarchical view of a river system. So Leroy Koch in '97 was talking about when we take a sample at a river site and we get the community within our sample, what's driving that pattern? What's driving the structure of that community? And he hypothesized that there are a series of filters that basically a community has to go through in order to determine what we see at the sample at the bottom. So we can start off with a regional pool of species at the top, and then it passes through, like I said, a series of filters before you end up with your community at the bottom. So we start off with filters, for example, at the watershed, or they call them watershed or basin filters. So these are things like biogeography, climate, and geology. So from your regional pool of species, compared with where you've taken your sample, some of the species within that regional pool wouldn't be adapted to, say, the climate that's experienced in that region, and so they've been removed from the pool. But the species that remain have managed to pass through that first filter. We go to the next scale down, which they call the valley or the reach filters. These are things like the slope, the land use within the catchment, the morphology of the site. And again, so your regional pool of species has been depleted slightly at this first filter, and again, it will pass through the second filter, and more species will be removed. Then we get down to the channel unit filters. So these are things like the substrate size, large woody debris at your site. And finally, the final filter is what Koch termed the microhabitat filters. So these are things directly at the point where you're taking your sample, so things like water depth, the flow velocity at the site, and also the particle size. And so the community that you're seeing here at the bottom is basically taken from that regional pool, passed through the filters, and he's hypothesized that this is what is driving the structure of the community. What GIS allows us to do is to be able to quantify these variables at these larger spatial scales, so these watershed scale and also this valley reach scale. Because when we go out and take a sample, often what we're quantifying, we're looking at the riparian vegetation, we're looking at substrate characteristics, we measure water depth and velocity, maybe channel width. But these are predominantly the smaller scale. And GIS allows us to take these large-scale variables and place them into the context with the smaller scale variables and provide that link to them. So how can we use these GIS tools to explore our watershed? So we have different software options. If you have access to this, is proprietary software by Esri, and that's ArcMap, which I'm sure many people are familiar with. Or an alternative is an open source version of a GIS software called Quantum GIS. And this is now readily accessible. It's very easy to use. It's more of a kind of a point and click interface than the traditional interface, which was more kind of code based. We also have different data sources. I've just pulled off a few. These are all data sources with openly accessible data. So there's the GeoBase site, which contains things like the National Hydrological Network. It contains a land cover layer and various other kind of watershed characteristic layers. GeoGratis here on the bottom left has just been kind of recently revamped and has a huge amount of geospatial information, not only that you could download and plug directly into a GIS software, but also maps and other documents available too. The MB Waters site the beta version has just been officially launched, and that's going to allow us within New Brunswick access to a myriad of spatial data within the province. Other provinces also have kind of a similar kind of geospatial data portal. For example, BC has the BC Geo Gateway, and again, these are brilliant sources for data, openly available data, and so you can start to understand what's going on in your local watershed. And finally, we have the Atlas of Canada data site, which has not only published maps, but you can also download some of the data that is behind those maps, again, to kind of put your watershed in the context of perhaps the province or territory that you're interested in, or even on the scale of Canada. So what tools can we use? So we've gone out, we've taken our sample on our stream network, and we've come back to, we've come and processed it, and now we're interested in understanding what's driving that structure of that community, what's going on within our watershed. So within both ArcMap and also QGIS and the other open source GIS software that are available, they have the same kind of tools available. So I kind of loosely grouped them. So the first ones we can look at are spatial tools, so we can go ahead and delineate the upstream watershed for our site. And from that we could extract things like catchment area, for example, or maybe the perimeter of your watershed. The second thing we can look at is maybe extracting values, so either for the entire watershed, or also you could delineate a sub-watershed, perhaps if you were interested in one particular portion of the watershed. You could extract the catchment area for that. We can also do a surface analysis, so by that I mean we can look at our kind of elevation profile, we can calculate slope, aspect, hillshade, and from there we can use something called zonal statistics, which basically allows us to extract information about those different layers. So we can extract things like average slope within the catchment, perhaps maximum slope if you're interested in that. And this is all using a digital elevation model as your base to calculate those layers, your elevation, your slope, and your aspect. You can also calculate stream network statistics. So in this example here, I've got the Strala stream order. So as long as you're following how you're supposed to calculate it, so you're supposed to have a 1 to 50,000 stream network, so everything is directly comparable, there's a stream order function within both programs that allows you to determine what the stream order is for your site, which saves you a lot of time from painstakingly going on a topo map and trying to count up how many first order streams you think you might have and determining what the stream order is below. We can also look at network tools. So if we think about the stream as a network, and we have, let's say, 3 samples within our watershed, we can look at the straight line distance between sites. Now that in itself is very interesting, say for example for benthic macroinvertebrates, because when they're in their adult stage, most of them are kind of winged insects, and so they may be using this kind of straight line distance between sites, between habitats, and you'd be able to quantify that. But something else that we're really interested in, because we work with streams and rivers, is the stream network distance, so the distance along the stream that you have, and so you can compare what the distance is between your base site here all the way up to this site here and up to this one here. And for example, that would be really useful for targeting the movement of particular taxa, and also perhaps understand where perhaps there's been some kind of contaminant input. You could map and understand how far the distance is along that network. We can also look at buffers. So this is where you have your sample, and then you can calculate a series of radial buffers going away from your sample point, because maybe you're interested in the land use change 100 meters from your site, 200 meters, 500 meters, or whatever you decide to determine the different intervals at. But we can also think about a kind of a buffer along the length of a reach. So rather than being a radial buffer, we can have a buffer that goes out adjacent distances from this stream, which is kind of more in parallel with what goes on within the kind of riparian zone. Most of the stream samples we see are being driven by what's going on with lateral movement and also this kind of longitudinal movement. And we can capture that within our kind of buffer there. And we can also intersect layers. So we can go to our watershed, collect a sample, bring it back and understand it. But what we want to understand is what's driving that, and maybe we have a land cover or a land use layer, and we can intersect the layers and try and understand, say, what the percentage of agriculture is within our watershed, what percentage of forestry, or if you have very detailed layers, different kinds of agricultural crops, whatever layers you have, you can intersect that and start to quantify what the percentage is within your watershed. So some different tools that are available. These are, again, are all freely available. ArcHydro is only compatible with ArcMap. So within ArcMap, under the spatial tools, there's a whole hydrology toolbox where you can use that to delineate your watershed. And that's a project and a workshop in itself, is working through how we actually do that. The tools here through ArcHydro are basically a kind of an add-on toolbox for ArcMap. And it uses the same processes as the base toolbox within ArcMap, but it has some useful features and useful kind of database management features to allow you to, for example, delineate multiple watersheds at once. We also have geospatial modeling environment, which again is another open source add-on. Again, this one is only compatible with ArcMap. So within ArcMap, some of the tools that you want to use are a little kind of convoluted to get to, or you have to do some scripting to be able to get the information that you want. So the people at spatialecology.com have developed basically a standalone program with a large number of tools that they found that would be particularly useful for kind of ecologists, people looking at watersheds, people who are interested in their environment, but just want to do kind of like a point, a click, and then fill in the information and run it, rather than doing the kind of raw scripting that you would have to do. A final tool here is called Ganggets, and this has been developed out of Dalhousie University in Nova Scotia. And this is just it's a standalone program that allows you to map the spatial properties and community properties of your samples. So you can map where your individual samples are taken, and then you can look at different properties of composition of your samples. So I have a couple of examples here. This is from the Bio2 project, which is currently ongoing up in Wood Buffalo National Park. And there's been a number of samples taken, and we can look at the composition of the samples in relation to both space, and then if you had them over different years, you can look at it across time. They also have an example here from the Sydney tar ponds, where they were looking at the composition of the different samples that they took. And again, this is from a large study that they did looking at the different composition of these samples. So basically within Ganggets, you can take the information that you have about your samples and you can produce these really easily interpretable diagrams to help you understand the spatial patterns within your samples. So I have two examples here of a GIS application in freshwater ecosystems from work that I've been involved in. So my first example is from the Atlantic Reference Condition Approach Model, which has been developed in conjunction with Environment Canada and CRI and also Protea in Italy. And I just want to briefly go over what a reference condition approach model is for those people who aren't quite familiar with it. So we can go to an area, a region, and a number of samples will have been taken, and we have to identify what we call reference sites. So these are sites with least impact, very low impact sites. We can then look at the benthic macroinvertebrate community within those samples, and we used cluster analysis to try and group out different community types. So in this example here, we've identified 3 different community types. So for each of those samples, we would delineate the upstream catchment, and then we would extract a whole suite of geospatial information about those samples. We would then kind of put that information, the geospatial information, into what's called a discriminant function model, which basically allows us to select the geospatial layers that best kind of represent the different clustering groups that we identified in the previous one. the really important thing to note here is that we're looking to use independent GIS variables. So these are things, when we're doing a reference condition approach model, that wouldn't have been affected by human activity. And then we can basically bring in a test site. So for that test site, we would extract the geospatial variables that we'd identified in the previous step. And we would use those geospatial variables to try and calculate basically the expected probability of the structure of the community based on if there be no impact whatsoever. Because as of yet, the test site, we don't know if it's impacted or unimpacted, and that's why we're running it through the model. So once we've calculated the expected probability of composition, in this case, we're saying, based on the geospatial variables, this particular sample is 30% type A, 45% type B, and 25% type C. So that means we can go back to our reference sites and we can extract a community that is based on 30% type A, 45% type B, and 25% type C. And that's what gives us our expected community for our test site. So in the absence of any kind of human impact at our test site, the community should look like this. And then what we do is we take the actual observed community sample that the observed community that we sampled at the test site, and we compare it to the expected community at that test site that we've just previously calculated. And basically any deviation from that expected community allows us to kind of assess the amount of impact at that test site. And so I've been involved in the Atlantic reference condition model, and this is where we took a whole series of samples across New Brunswick and Nova Scotia, PEI, and Newfoundland. And we had 128 what we call training samples, which is what we used to build our model. We had 42 validation samples. So we use those to just validate that the model is correct and also assess any errors associated with the model. So we had 170 reference sites, and each of these have been collected at a unique location. So for each of those sites, we delineated the upstream catchment, and we extracted a series of geospatial variables. Now, all of these variables were selected so that they were nationally consistent, with the idea that if we could successfully develop an approach here in Atlantic Canada, then maybe this method could be kind of applied in different regions of Canada. So we looked at things like topography, so with that we're looking at elevation, slope, and aspect. Climate, so we were looking at long-term climate records provided through NRCan. We looked at bedrock geology, soils, and we also looked at hydrography, so that's looking at kind of network descriptors. And when we ran the discriminant function models with the data that we had, we selected climate variables and also bedrock geology variables as being the drivers of the community, or the kind of discriminators of the community clusters that we saw in the previous example. Now when you run your model, we can actually bring it into Ganggets, which is another add-on for Ganggets. We can integrate the results with the software. And this allows us to visualize the model output. Because often what happens is you get a value for your site, but you're unsure about, how is that in relation to, let's say, the neighboring watershed, or how is it in relation to a watershed in another province? Well, Ganggets will allow you to map your samples and map your outputs, and so you can look at that kind of regional context. And so in this example here, we have our samples that we use to build the model on our test sites, and then this is just an example of the output. And so when we do that observed data and compare it with the expected data, we can calculate a series of indices, and these are the outputs from the indices. And so it allows you to visualize your data. The second example here I have is the thermal mapping project, and I know that this was actually touched upon in the first webinar of this series. For this part of the example, it's going to be looking at data that was collected within the Cairns River, so within the Miramichi watershed as opposed to the Restigouche. But just briefly, for those of you who aren't familiar with it, a series of reaches of both the Miramichi and the Restigouche rivers were flown using infrared cameras. And these allowed us to quantify the water temperatures along these reaches with a view that we were trying to map the habitat, the cool water habitat, identify the cool water habitats, which is so important for a number of species within these systems, particularly with the ever kind of increasing water temperatures that we're seeing. And so basically what it allows us to do is you get images like this, where you can see this is the main channel running along here, and then we have cool water sources coming in. For example, here we have Esty Brook and also an unnamed tributary, and it allows us to map how far along the reach the influence of these cool water refugia are. And it also allows us to identify cool water refugia that aren't necessarily on a map. So it might be something that isn't as clear as, you know, Esty Brook there. It may be an individual seep coming in here, which unless you were walking along the kind of length of the reach and had your hand in the water or a thermometer, then you wouldn't have known it was there. And so if we can map these cool water refugia, then that allows us to better manage and protect these cool water refugia and understand how important they are. So this is all within a GIS system, and then we took along the Cairns River, we went and we identified all of the thermal refugia along the reach of the Cairns that had been flown, and they're kind of represented by these grey dots here on the map on the right. And then what we could do is we, again, we delineated the upstream catchments for each of these kind of tributaries and sub-tributaries that were identified. And then we could extract, again, using geospatial information, land use, land cover, geology, soils, and a suite of different kind of hydrography and catchment descriptor variables to try and see if we could get a handle on what was driving these cool water refugia, what were the variables that were most important in determining the presence of a cold water refugia at that point. Because the idea then is that you can go to other parts and you can use the same geospatial information to try and, you know, predict whether there will be cold water refugia present. Because flying the infrared cameras is a very costly process, and so if we can then start to use it in a modeling approach to do that, then, you know, we can have a wider spread applicability of the method. This is also building on the same project. This was again from Dr. Tommy Lensari's work, using this same thermal infrared mapping output. So they have the map, and then they went and tagged a series of fish, salmon, within, this is along the Little Southwest Miramichi, and they wanted to see how these fish would use the known cold water refugia during a thermal stress event. Because for a lot of the, you know, the salmon within the system, once the water temperature goes above a certain threshold, then the fish suffer stress, and they may alter their behavior and move to these cold water refugia. So before the thermal stress event, each of these dots here represents an individual fish. And then we have two time periods. We have during the thermal stress event and then after the thermal stress event. So during the thermal stress event, the majority of the fish move to cool water refugia. So Catamaran Brook coming in here is a cool water habitat here, and it has, in effect, kind of going downstream. So those fish have abandoned their regular habitat and have moved to the cool water during this high thermal stress event. After the thermal stress event, the fish redisperse either to their previous habitats or near to their previous habitats. So I think this is a very nice example of kind of integrating GIS with the behavioral patterns of these fish, because we can have a whole lot of information presented in one figure. We have the thermal imaging, we have information about the sites, and then we can also map onto that movement of the individual taxa to be able to present our results. So just briefly there, in summary, I've gone over a very brief review of mapping history and also the different types of maps that we might be interested in. I've introduced some tools and some software and some data sources that you can access and you can use in your work. And I've also just given you two kind of brief applications of GIS in freshwater ecosystems. So I'm just going to leave you here with, these are some of the useful open source links that I referred to during the talk. So there's the Quantum GIS software, which again, as I said, it is very readily accessible for those who perhaps don't have access to Esri's ArcMap software. There's also the 4 main data sources that I talked about, but again, you know, for each province and territory, they also have their own kind of geospatial data warehouse. And finally, there I have the links to those 3 tools that I mentioned as well. So thanks very much for listening.
Thank you, Wendy. That was absolutely excellent. A lot of really great information there, and thanks for including all of those links at the end. For everyone who might be frantically writing these down, we will be posting these on our website afterwards, so you'll be able to have access to the presentation there as well. We will now open the floor to questions. So again, you can raise your hand or you can type in your question and we'll read it aloud. So Michelle, are you seeing any questions on your end?
No. Oh, one just showed up. Just a second. Okay, a couple are showing up. So, first one is from Susan Stanley. She said, what software would you recommend for watershed delineations?
I would, well, if you have access to Esri's ArcMap, then you could use that. But equally, Quantum GIS, the open source software, it's very easy to use. The watershed delineation approach is exactly the same as Esri's ArcMap software. So it would depend on kind of the resources. Esri's ArcMap, unless you're working with a large organization, then likely you wouldn't have access to that because it's rather expensive. But Quantum GIS is an equally good alternative to Esri's ArcMap.
I'm just going to put a plug in here, shameless plug. So Wendy is currently developing aquatic techniques or tools for GIS for the CRI, and we're running a test run in early December. And sorry, folks, if you're just hearing about this now, we've already filled that up. It was just a limited number of people, so we're going to run that, and then when we get feedback from participants, we'll run that in the new year whenever Wendy is willing to do so and when demand permits. And that will be focusing on using the open source software like Quantum GIS and linking to R and stuff like that. So maybe folks that are interested, you can send me an email, m.gray@unb.ca, just to indicate your interest. There will be a fee associated with it for cost recovery, but we'll be sending out notices in the new year. Okay, on to the next question. Scott Wilson says, how did you collect the thermal imaging data?
So the thermal imagery was basically they attached a thermal imaging camera to the bottom of a helicopter, and then it also had on a very accurate GPS unit, and so they flew reaches of the streams and rivers at different heights depending on kind of the width of the river, and using the accurate kind of GPS output, they were able to geolocate each of the thermal images that were collected from the camera, and then these were all kind of merged together to form the images that you saw back there. And that's how it works. So it's basically a combination of thermal imagery and GPS. And then there was also some ground truthing too, so they placed thermal loggers at various points along the streams which were logging for kind of the whole summer season. And then they would know when the helicopter or plane had flown above with the camera on, and so they could just basically do an error check. Is the thermal imaging results that we're seeing, does that match with what we know is happening in the stream based on our logger output? Hopefully that answers it.
Hopefully. Oh, he says thanks, so I think you did a good job. Next question is from Andrea Kirkwood. How is the geospatial data extracted and used in statistical analyses such as ordinations?
So basically what you can do is you have your various geospatial layers. Let's say we take something like land cover. We've delineated our catchment. We can then use what's called the intersect tool, and so we can for our catchment we can calculate, I don't know, the percentage of the different types of land cover within our catchment. And that's what becomes the basis for your, let's say, your ordination or your analysis. So you would have for each sample, your sample site, you'd then have a series of columns representing the data that you've extracted from the GIS. So you may have catchment area, you may have average slope, and then you may have the different percentages of your land cover. basically the problem with GIS is that you can create a lot of data in a very short amount of time. And actually one of the problems comes is how do you deal with that in your analysis, because you can have far too many environmental variables to try and fit to your community data. And so what I encourage people to do is you go ahead, you extract the variables, but then you go through a data reduction kind of process to reduce the number of variables that you have, because inevitably a large number of them will be highly correlated with each other. And so you can do that in a data reduction method.
She says, that's exactly what I was wondering. Thanks.
Okay.
Okay, next one is from Nancy. Hopefully I'm saying last name's right, Serediak. The examples seem to reflect big systems. Are there tools available for smaller systems where available DEMs aren't of a fine enough resolution to allow standard delineation tools to work well?
Yes. Basically, you can do GIS on whatever scale you're looking at. The examples I provided are kind of reflect my work, which is looking at this kind of larger catchment scale kind of processes. But yes, you can look at smaller scales. Often, the readily accessible digital elevation models within Canada are at like a 30-meter resolution. In some areas, they've actually got far finer resolution. One thing you can do is you can take your DEM, your digital elevation model, you can kind of rescale it down to a smaller resolution. Of course, there's error associated with that, so it would depend on how fine you were looking to go. But that's certainly one thing you can do. And all these tools, these will all work at whatever scale that you're looking at. I hope that helps.
So, Susan Stanley has another question. What elevation, oh, she says thanks, so I think that's good. Susan Stanley says, what elevation data set did you use, and did you have to complete any pre-processing to get accurate delineations?
Yes, that's something that I didn't touch on, and it's something that within the course, the workshop that we're going to be doing in December and again in the spring, that we'll certainly focus on. So the first thing when you get a digital elevation model is the older digital elevation models were based on aerial photographs. And so what happens is that they're kind of mosaic together, so you end up with a whole sheet of what your digital elevation model looks like. But one of the problems is when you come to delineate a watershed is sometimes those joins between those individual photographs aren't great. And so you end up with a straight line in your watershed, which of course makes no sense whatsoever in the environment. So if you're working with older digital elevation models, then you go through, it's like a process of almost smoothing between those gaps and the joins between the aerial photographs. If you're working with newer data, so satellite imagery which is being converted to a digital elevation model, you don't tend to have this kind of join error, but you do get elevation errors and you get kind of quirky pixels almost where there's no data there, so they're kind of like dud pixels within the data set, or where the value doesn't make sense. So before I do any kind of analysis and extraction on any of my digital elevation models, I go through a series of checks and balances almost, going through and making sure that the data makes sense. If there are any dud pixels, then depending on how many there are within an area, we can fill them in if we're comfortable with that, or if there's too many, then we can exclude that area. The second part of your question in terms of the correct watershed delineation, what I would tend to do is if I have a known stream network and using the stream network through GeoBase, for example, the National Hydrological Network, is a great place to start. If you're comfortable and happy with your stream network and you're not having to define one from scratch, then you can take that stream network and you can do what's called burning it into the DEM. Basically, you're kind of taking a stream network and you're forcing the DEM to take that stream network and you burn it. So it's almost like the water's flowing along and then it falls into a big dark chasm. But it's just an artificial chasm that you forced so that when you come to delineate your watershed, hopefully the water flows and then falls down and then will pool at the base of your watershed, and it makes for accurate watershed delineations. Now it doesn't always work, but even in kind of flatter regions of the country, if you burn the network into the digital elevation model, then you get far more accurate results in terms of your watersheds.
She says thanks. Okay, and then Zachary and this is where I'm going to try hard. Sarah Bahenda says, which GIS tools can we use to delineate upstream area to station in river?
Upstream area to station, so you just want to calculate the distance or the actual area, sorry.
I'll just wait. Zachary, could you type that in? I'll wait for Zachary's response. There's one more question waiting from Donald Bowes. What is the relationship between the accuracy of watershed elevation data versus the size of a watershed? For example, is a geo-based DEM effective for estimating a watershed less than 100 hectares?
This comes back to that earlier question about, whether you can use these GIS tools for these smaller watersheds. You've got to be comfortable with the data that you're using. Again, if you're using the geo-based digital elevation model, then you're going to be probably looking at a 30-meter one. So when you're looking at a smaller watershed, you have to be careful because otherwise you're going to end up with an unrealistic watershed. So that's where you can kind of rescale the digital elevation model, and so you could, say, end up with a 10-meter by 10-meter pixel. But again, that comes with potential error, and if people are interested, I can send links to how you go about and do that. A lot of regions now are starting to develop these kind of more fine-scale digital elevation models. So it is possible to do. So if the data isn't there, then you can certainly rescale it yourself.
Okay, so Zachary responded with an example, for an example in a watershed, % of forest. So he had said, what GIS tools can we use to delineate upstream area in Station River? So I guess
so you just want to calculate the percentage of forest, say, within your watershed. Is that? So that's using, you would just use the intersect tool in either Quantum GIS or in ArcMap, and you would have your zone or your area would be your watershed that you've delineated, and then your layer would be, in this case, forestry, and then you can extract it, and it will extract the total area of, let's say, forestry within your upstream catchment. Basically, think about it almost like a cookie cutter. So your watershed is like the cookie cutter, and then you're going out and cutting out the information from the layers.
Okay. Oh, what? That's right. Nancy says one more pesky question. That's okay, Nancy. It's all right.
No, ask away.
Exactly. So this is Nancy again. One more pesky question. When dealing with seepage systems, where should the pour point be placed if there is no defined outflow?
Yes, that's where we start to have problems with the watershed delineation. There are ways that you can almost trick the the model to be able to account for that. Yes, it's very tricky, but there are literature out there, and again, I could send links if you want on how to deal with that. But it's basically you can kind of trick the software into where it should be and where it shouldn't be. Oh, sorry, just adding on to that, there is, with the ArcHydro was with the stream network delineation, if people are interested in kind of delineating groundwater, etc., the same people that developed ArcHydro have also developed a groundwater add-on tool so you can model through flow and things like that if people are interested. And again, it's all available through the ArcHydro team.
Nancy replied by saying tricks are very welcome.
Yes, there's a lot of tricks.
I don't have any more scanning right now, so we'll just give it a couple seconds in case. I believe that's it.
Okay. That was absolutely excellent. Thank you so much, Wendy. I think judging by the number of questions that you received and the depth of the questions, that was very well received.
Thank you very much for inviting me to give a talk.
Well, thank you so much for speaking. And so just a reminder to everyone, there was a lot of information there, but we will be posting it on both the CRI and the Atlantic Salmon Conservation Foundation websites. I'd just like to put in a little plug for our next series of webinars. So our next presentation is going to be Stream Restoration Hydraulics, an introduction on November 6th with Bob Newberry. And for those of you who haven't heard Bob speak before, you're definitely in for a treat if you sign up for this one. Bob is really renowned in the world of hydraulics, and we're really lucky to have him presenting. So that one again is on November 6th, and the following webinar will be Daniel Cassie on November 20th, and he's going to be speaking on the importance of hydrology and river hydraulics for salmon management. And so this is another great webinar that we'll be looking forward to. A reminder about this one, though, it is going to be in French. So registration and a listing of all the webinars that are coming up on this series are on both the ASCF and the CRI website. So you're welcome to check those out and to join up there. So that's everything for today. Thank you so much, everyone, for participating, and we'll hope that you'll join us for another webinar very soon.